Next topic is from instrument to insight, building the Agentic pair lab. The speaker is going to be Jeff Morgan, who is Managing Director in Deloitte's Life Science R&D group, with practice with more than 20 years of experience helping life sciences organizations modernize R&D through data analytics and AI. And I believe he's going to be joined by one of his colleagues, Prerna Patil.
Thanks, everyone. And Vinny, thanks for the invite. The conversations yesterday and some today kind of really underscores a key premise that I have around use of AI apps. And it's not going to reach its full potential unless we have that contextualized data foundation that we all have been talking about here. So I'm going to spend a few minutes talking about how AI automation and connected data can create a fair lab that allows you to make smarter decisions in the lab. And then we were talking about how do we articulate the value of ASM? And there's conversations at the end of the day yesterday and some today. And I kind of took a step back and I was looking at the work that Deloitte does in our R&D practice across essentially all the top major pharmaceutical companies, and we span all aspects of R&D. Most of the work we do is grounded in trying to improve R&D productivity for our clients, right? And if you look at R&D productivity over the last 15 years, it's been on a constant decline, right? There was a little uptick about 2 or 3 years ago with GLP-1s, but for the most part, it's been on a decline. So clients are investing in building out capabilities to really reverse that trend. And this is a problem you can't buy your way out of in the sense that a lot of companies are overpaying for molecules in late stages. So you really need to reinvigorate your internal research engine. So to me, that's where these worlds come together. Right? What the investments we see in the labs today are really around laying that data foundation that can enable those smarter decisions in the labs, and ASM could play an important role in making that data usable at scale in the labs to really drive that innovation and increase that productivity. So I'm going to talk a little bit about what we call our Lab of the Future solutions for the next couple of minutes. So when you hear the term Lab of the Future, everyone's familiar with it. And most people picture robotics, fully automated labs running 24/7. And all that stuff is true and it's important, but to me, it's not the core it's not the core idea of these labs, right? It's to have what we call, integrated, well-connected data. That's the scientific backbone that can enable faster, more intelligent decisions to enable AI at scale and discovery, right? So that's really why you see organizations investing in this space. And when I think about the vision for Lab of the Future, it's really to kind of close the loop between the digital design and the physical experimentation, right? So you think about those two worlds today in the design, make, test, analyze process. There's a lot of manual handoffs and friction within the process. And what happens there is it's slow, there's room for error, and you lose context, right? So the opportunity is to create a more connected ecosystem between those two labs where you have high quality experimental data that can improve models. And then those models could then inform the next round of experiments, right? So you create this learning loop. And it's not about automating any one step or speeding up any one step. It's really around making a faster learning cycle within the lab environment. That's really what's going to drive that innovation for the pharma industry. So over the last, say, 18 months, we've been partnering with AWS to develop what we call our Lab of the Future suite of solutions. So this is at the bottom is design principles. So it's open, modular, cloud native. So a lot of what's built here is AWS native. So for the organizations that leverage AWS, which is honestly the majority of the pharma industry, these are all kind of cloud native to AWS. They're scalable, standards-based. We use the ASM model, and there's agentic capabilities throughout. And we use the phrase at the top there, instrument to insight lifecycle. So I'll walk you through this picture now and show you how this going from the instruments to the scientific insight at the end, how we can remove friction, right, and enable friction of science. So on the left here is a representation of the instruments that were being in labs, right? These are not all the ones, but flow cytometry, digital pathology, imaging, et cetera. The first challenge is getting that data into the cloud, right? So that's where we built the data mover monitor, which is a fast configurable way to automate moving data once it's generated at the instrument or on-prem into the cloud, right? It can do this at scale, and there's no human intervention. The solution is constantly watching for files to be completed, and then it moves those files into the cloud. Up at the top left-hand corner of the big box over there, we have a control tower, which is a nice UI where you could actually see all your instruments in your fleet, right? You could see what instruments are connected. You can see what data is moving off of each instrument. You can see if there's been any issues, right? So you can monitor your pipelines and your fleet of instruments. You can also see, which instruments aren't being used as much. So you can kind of maximize your investments in those instruments. So once the data is landing in S3 and AWS, this is where we start to turn that raw data into something usable for the scientists with our parsing capability on top there. There's a lot of talk today or yesterday and today around developing parsers with AI, and we do that. What's a little bit different here is not only can we develop parsers with AI, and I'll go into more detail in a few slides about this, but we can also monitor for drift and self-heal those parsers. And then we contextualize that data with metadata from ELNs and LIMS systems. We can do a bidirectional push or pull from those as well, right? So that's where they create those data products that the scientists can use. And then on the far right, once those data products are developed, you can automatically populate a data catalog so that it can be governed and visible to the organization and scientists. Then the bottom right there is kind of where the outcome that matters to the scientists happens, right? They have a workbench where they have all their tools, and they have a chat user interface where they can start to ask questions to see what data is available to answer their specific research question. And we'll show you this in a demo, but I heard some comments yesterday around, wouldn't it be nice if we just had a chat interface? So we'll show you what that could look like, where scientists could go and put in their research question, and it'll start to search across the various systems to find what data is available to address those research questions, and it could actually even start to standardize and combine that data. So this is all in an effort, right, to reduce the time scientists take around, we saw it in the CPI presentation, right? There's something like 70% of their time is spent on administrative stuff, looking for data engineering, right? The goal of this is to remove the friction in the system so that those scientists can spend more time on science that they were actually hired to do, and not on those other activities, right? And ultimately, they want to be able to find higher quality, differentiated targets and candidates, and reduce cycle times. So I'm going to talk about a couple friction points that we see in that kind of instrument to insight journey within most clients, right? And how we're kind of tackling this with agents. So two friction points that come up over and over again is, first one's around parser development and maintenance, right? So parser logic is very bespoke and maintenance is on, right? So as files change, parsers can break, which typically requires manual rework, right? Which slows the whole process down and slows the process of getting usable data to your scientists' hands. The second pain point we see a lot across the industry is around assembling data for analysis. And I kind of mentioned it before, and from the CPI report that your scientists are spending a lot of time looking for data and combining data to answer their research questions, right? A lot of manual steps go into that, right? So these are two friction points that we see again and again across the industry, and we've been able to address with agentic capabilities. So first I'll talk about a parser agent. So it's got three main capabilities to it, right? First, you could build a parser with an agent, right? So it'll once you onboard a new instrument, you can build that parser. It'll automatically generate tests and register that parser in our parser library, right? So now the agent can refer to that parser if a similar instrument comes on and pull the parser. It converts the outputs into ASM standard outputs for the scientist. The second part is around that drift detection. And I'll go into more detail around this, but it can automatically detect drift in the schema, in the structure. And then there's also, it'll create a drift score to determine what action might be needed if that parser drift or if the file drifted. And then the third piece is really around, it could rebuild the parser if it needs to be rebuilt. So it'll automatically trigger a parser rebuild. And then there's a human in a loop validation part to this where humans could come in and check and ensure that everything looks fine before putting it into use. So it's really a self-healing capability now for these parsers. So talking about the parser drift, right? So it does three checks, right? So it does a structural check. There's the schema match. Does the file match expected schema? Are all the required fields present? Is the format correct? It does the semantic evaluation. Do these values still make sense in context? Have any of the units, codes, or value names changed? Will the output still be interpreted? Then finally, there's a business rules check, right? You could create client-specific rules and make sure they're satisfied. Instrument versions and value ranges and process expectations met. And or any of the other rules that we defined in the skills file still being honored. So it goes through all these checks and then it'll create that weighted drift score that gets used to determine the next action of the agent. For that second pain point that I talked about, where scientists have to discover data and assemble data for their analysis, we've created what we call our dynamic dataset builder. So you can think about this as a conversational agent, and we'll show you this in a demo in a minute or so. It's a conversational agent where you could input your research question, then it will go search for data using internal sources and scientific concepts. It'll show you what data it found. You can validate what that the data sources are correct, and then it helps you assemble the data set. It'll start to auto map the identifiers, standardized units, and create a cohesive data set that the scientists can use to answer their question. And then from there, they go answer their question with the tool they want. They could export the data into the tool that they want to use. If they want to reuse this data or share it with other colleagues, they could publish it to a data catalog or share it with them. So it's really kind of compressing that time it takes to go from research question to answer. So with that, Prerna, are you on the line?
Yes. All right. Hi, everyone. Nice to meet you all. So as Jeff was talking, right, so this is our workbench where scientists have different tools that they can utilize for their daily workflow. So we are going to get into the dataset builder. So as we've seen, researchers, across the pharma organizations generate or conduct a lot of experiments throughout the year, right? And as they're conducting these experiments, it generates a lot of data, and this data is just stored in different systems. And when researchers would want to reuse that data, they would have to kind of go in these systems, figure out what system it lives in. They would have to kind of stitch the data, clean the data, figure out if it's compatible for their reuse, and then come up with, create that data set that they would like to utilize. And this will probably take them weeks to months sometimes. And then also they require specialized skills to do that. So to to answer those questions for researchers in a natural language, we have built this dataset builder, which can do this, we wouldn't say kind of in minutes, right? Like based on the data that you have, it'll allow researchers to ask those questions and then also build a data set that they can reuse for other purposes. So to begin this, what we are going to do is we are going to start with a question that the scientist has. And then what this is basically, this is LARC2 is a protein which is linked to Parkinson's disease. And this G2019 is a mutation which is kind of makes the faulty version of itself. So the scientist is asking, is all of the historical data, how does this protein behave differently between the normal version and the disease version, right? So that's what the scientist is asking. And then what the agent does here, before it really goes and searches for all of the studies within the organization, it's actually asking, before I search for it, it's going to ask targeted question, right? So this is particularly the first question it's asking, do you have any cell line preferences? So all the agent is going to, not just go ahead and search, but it's going to understand what the user really wants before it can suggest and recommend and build that data set, right? So here you can see like from the first questions, this is what is understood. And then it's like, okay, for me to, further narrow down the stuff that I can really build for you, I have this first question. Do you have any cell line preference, right? So here we are going to say any cell line is acceptable. So it's thinking, and then it's going to now understand that. And then it also has another question, right? Do you have any expression systems or any preference for that? So it's also bringing any suggestions during that time as part of that. Here we are going to say any expression system is fine. And then what the agent does now is it's confirming the full scope of what it understood, right? It's not just going and searching again. It's understood that based on the questions it's initially asking, this is what I've understood and this is what I'm going to do. So at this point, either the scientist can go ahead and confirm the search, or you can adjust, right? So if I want to say, when I had initially asked something, I may want to adjust it. So I can say, hey, I might want to just go ahead and adjust a little bit. So it's thinking, and then it's asking, what do you want to adjust, right? From these things, you have a target, you have a modality, cell line, what do you want to change? So in this case, I'm going to say, prioritize the neuronal cells, cell lines, and keep all of the cell lines that were in scope, right? Just add these additional ones. And just wanted to call out here. So as we are building and asking questions to this agent, you can see on the right-hand side that we are also populating the data set summary is getting populated. So at each checkpoint, it's also populating the summary at the right-hand side. So at the end, if you'd like to see what was the goal, what was the scope, you have it readily available on the right-hand side. So here, I'm going to go ahead and respond to the agent. So it's thinking. And then it's going to, provide the updated scope, right? We had already provided the scope for it, it understood, but now it's going to, since we updated and adjusted the scope a little bit, it's saying, okay, this is what I understood. What do you want me to do, right? So I'm going to say we confirm, and now go ahead and search for the studies, right? So what it's going to do now is, and as you'll see, that it's going to come up with a table of studies. Is the studies that it's looking at in this case, we have a data set that we have built for 5,200 studies, and it's looking across that because these studies that you will see are of different types, different disease mutations, different lab setup, different instrument, and these studies have been conducted over a different period of time, right? So the job of the agent is to really look through these studies within the data store and figure out the studies that match the question that the user is asking, right? So here, the agent is responding. Here are the 5 studies with the question that the data is available. And this is laid out as nice table, which shows you the studies, the ELN ID, what year it came from, and then the different attributes here. And then you can see on the right-hand side, we have two columns here, which is the match and the risk. So this match is basically, it's telling us how good this data is, right? How relevant it is to the question that the scientist has asked. And the risk is telling us how clean it is, right? So what the scientist or what we'll be looking for is all the high plus the low studies, right? So it's giving the user that confidence. So anything that has a high and a low risk ones are the ones that that it's brought out. And then there are some notes additional to that. And down below, you can see like it's also recommending. I found these studies, but what are the actual recommendations? So it's providing, like, if you want to combine it, this is the core, and it's providing the rationale with the mutation and the protein that we want. What is the best set that you want to have? So it's recommending some of those. And then here it's also saying the adjusting that I did for the scope, it's saying the neuronal cells studies, the data has not been ingested for these studies, right? So even if you wanted to include that, the data is not available. So it cannot be a part of this current build. So it's also gone and checked that, and it's letting us know all of this. So in this case, I'm going to say, yeah, I like these studies that you came up with, and then I'm going to accept all the changes. And this will take a couple minutes, not minutes, a few seconds, sorry, because it's looking through a lot of data here. So the next step that it's going to do is, here it gave us the top 5 studies. So now what it's doing is it's doing a metadata alignment check across those studies. Does the metadata for these studies make sense for the question we are asking and for the data set that we want to build, right? So it's saying this is a nice table that it's laid out. And for these different attributes, it's giving us the check across the matrix for this metadata. And then it's providing a harmonization plan. So for some of the ones, it's saying that the entire metadata does not match, right? Obviously. But how would you want to change that and harmonize that data? So you'd have that data set that you can utilize for your research. So it's providing this plan for harmonizing it. And the mixed in this case means like every single particular study may not have this specific or same attributes, right? So it's providing that context here, and then it's saying what is the action required for it, right? And then it's also saying that all of these, even though we found out that there are mixed attributes in there, they are all annotable and they're manageable, right? So there's nothing to worry about. So we'll be able to harmonize that and then manage these mixed attributes in there. So in this case, we are going to say, okay, this is great. I like what I see. I'm going to approve. And then it's going to go ahead and look at, and now it's looked at the metadata. Now it's going to, in the next step, it's going to look at the actual data associated to these studies, right? So the metadata sits somewhere else in a specific catalog, for example. Now it's going to look at the schema that's linked to these specific studies. So here you can see like it's done a schema check, and it's come across some of the issues that it found in the schema, right? So the first one here, it's saying that, it's found an issue with this. And it's saying that we would like to, it would be best to quarantine this data set because it's found some data quality issue in there. Okay. And then the second one is, a medium risk. We've found 2 issues, right? And what would you want to do, right? For this, it's also providing resolutions at each of this. So here it's saying it's quarantine. You can quarantine this data set and not include in the final data set. And then this one is proposing that we flag these rows and add additional column for this one, right, in the data set. So I'm going to say, yeah, approve all the schema resolutions. I like what I see. And now it's going to show me the actual data for it. So it's confirming what we just did, right, the normalization. And what it does is it's going to give me in this data set inspector, we're able to see the sample rows before I can really, before the agent can really pull out the actual data that we'd want to use. So when I click on this data inspector, these are like 200 rows of the sample for those studies that we had finalized in the chat. And this is where I can really look at the data, scroll through and see the actual data for each of the studies. And then we have the filters or the core attributes on the right-hand side. I can, for example, if I don't want the genotype or I don't want any of the acquisition mode, I'm able to check and uncheck some of these attributes, which I want to include in my study on my data set. And then I can apply the selection. So this is being taken care of by the agent. Now it's going to start building the data set. So it's thinking. Before it starts building, it's going to give me this summary of all of the things that that was confirmed during the chat. So it's just nicely lists down. So it's going to build this, first data set, which is a comparable protocol set, and the second is a broad set, which includes additional studies that it found as part of the additional metadata that we are looking at. So now we can look at this QC summary, and then I can go ahead and approve and export. So what it does is it's going to export this. If we want, we can export this to a CSV or in a notebook, a Jupyter notebook, and then that can be then utilized by the scientists for other research purposes. So this is kind of what we just saw is this natural language way, a conversational way for scientists to come in and build the data set and ask research questions of the data before they can really start building. This would have taken like some scientists, looking into different systems and figuring out what data is available, then try to see if it's compatible, if you have the right metadata, and then try to stitch it together. And now this is done kind of in a conversation, right? And then once we are able to get this, we are also able to save this as a recipe. So let's say in the future, if there's somebody else who would like to utilize this, we can also save this as a recipe in the catalog and will be available for future uses.
Thank you, Prerna.